सबको नमस्कार कोएलिशन फॉर जीएम फ्री इंडिया की तरफ से वी वेलकम ऑल ऑफ यू ऑन बिहाफ ऑफ कोएलिशन फॉर जीएम फ्री इंडिया दिस इज गोइंग टू बी ए वेरी बेसिक training course on understanding this genetic engineering, genetic editing, especially. Beyond genetic engineering now, there is a genetic addition kind of
thing. I think all of us would know that all of us have something called genes, chromosomes. So it goes from generations to generations, and anything that we have, any traits or features that we inherit are inherited through these chromosomes that are passed from our parents. These things probably we know. So these chromosomes are made of something called DNA, which is deoxyribonucleic acid. So it's a kind of a chemical. So just like you say carbohydrate, you have starch, you have sugar. So all these things are there. So these are just chemical. I mean, name of that chemical is called DNA. So among the nucleic acids, we have DNA and RNA. So this you can compare to, like for example, a cookbook where you have all recipes. From there, we need one item that you want to prepare, and you just take that sheet out. And that sheet you take to wherever you want to cook, and then you make some dish. So that is what happens when one protein has to be made. So now, again, one more thing. What is a protein? So proteins are those things which actually do all the work. So again, another analogy for the same thing is, suppose you have a king who is in a palace. He gets to know about some place being affected by rainfall. He wouldn't actually go there and look at it. He would get the message from somebody. The moment that information comes, the king would probably tell the minister, oh, please send all the relief material immediately. And the minister would take that information that the king would have said in a colloquial language, put it up as a government order or something, and then get the work done. So the work would be done by all the workers. So those workers are analogous to the proteins that we have. So this process where the king spoke to the minister, so the DNA would have all the information, it would convey that to a minister. That language is going to be the same. So that process, we call it as transcription. So that's like, suppose I have a message like don't disturb in DNA. Suppose the minister takes it as the same language, but then transliteration of it. And then when it gets conveyed as a government order, it's another completely different official language. So this is what is transcription and translation. I have a small video which would take us into the cell and give us perspective about what is the dimension that we are dealing with. So when I say about a palace or a king, our imagination of what we are probably looking at is too big. So now let us just look at this to understand what is the level of organisms that we are talking about, or the cell that we talk about. So suppose I get into a cellular level. So I have the body and then the organs. Inside those organs, I go further inside, I have cells. Suppose you take the liver cell. Suppose you get into the liver cell now. This would have lots of organelles. So just like we have organs, inside a cell, every function is delocalized in a particular place. So all this DNA is present inside a place called the nucleus. And the DNA is just not floating around like that. It is organized into chromosomes. And human beings have 23 pairs. So this pairs, let us just remember why we have to be remembering about pairs. It's not just one copy. We have 2 copies of every chromosome. So one is from the father and one is from the mother. Each of these chromosomes, if you see, is a very, very long stretch of DNA, deoxyribonucleic acid. It is packed this well. So to put it in perspective, one cell is not even as big as 2 lines of your fingerprint. Nucleus is even smaller. In that space, in a human chromosome, in a human cell, you have 2 meters packed inside that space. That is the amount of DNA we have. So these DNAs are written in a language which has only 4 alphabets. A, C, G, and T. So A always combines with T, G always combines with C. The stretch of these alphabets, we are learning it as alphabets, but these are these chemicals again. A stretch of these chemicals, are called a gene. So for every protein, we would have a gene. So if we make books of every protein that we can make from a human chromosome, we would have so many 120 million books. So that is the amount of information we have in the genetic material. So I hope this would have given us a perspective about what we are dealing with. When human cell is this small, everything else is smaller than that. So all the other cells are smaller. What we are dealing with is even smaller than that. So what happens inside this nucleus is, so like the palace of the king, we have everything coded in the DNA. The messenger would take the message, bring it out to this place called the cytoplasm. So that is the place outside the nucleus. Before bringing it out, in all higher organisms like animals and plants, the gene is not like a single message. It is coded. So again, taking the example of kings, they would usually give coded messages when they send messages through a messenger. So it is coded. This is similar to the word, sentence jumble. Suppose this is the jumble, to I jump like. What would happen is the messenger would then, reorganize that message, would make the jumble in such a way that some of the bits are removed and some are rearranged, so that you get a meaningful sentence which would become I like to jump. So for bringing these, there are enzymes which would cut, bring it here, reorganize. So these mechanisms happen in this place in the mRNA. After that editing happens, the mRNA would come out, then a protein would be synthesized. This is what would happen. So here I just wanted to point out that for one gene's expression, it's not that just one copy of the gene has just comes out. It's not like you taking one sheet and reading some paragraph. A lot of things are interconnectedly involved. It's like a set of dominoes that you stack up. You just hit one and everything will fall, the whole pile will fall. So altering anything would have a cascading effect. So when we look at these cells, we have two types. We have these which are higher organisms, plants, animals, and in all of them, the like what we saw in the video, you would have places where specific things would happen. So the whole DNA or the chromosomal DNA is packed inside a nucleus. Only the mRNA would come out. But in organisms like bacteria, you find that the chromosomal DNA itself is spread out throughout the cell. For all practical purposes, the whole DNA would be expressed all the time. Proteins would be produced from it and so on. So what the bacteria started doing is, it started keeping aside that DNA which is not required for everyday living. Like for example, in our situation, only when it rains we would take the umbrella out. Otherwise, we don't keep it open all the time. We would probably fold it and keep it aside. Similarly, this bacteria started removing chunks of DNA which were not required for everyday living. Like for example, an antibiotic resistance gene would not be required all the time. So it started putting it aside as an extra chromosomal DNA, and we call it plasmid. So whenever there was an antibiotic stress, it would start expressing that particular gene. So this was what happened in bacteria. When they discovered that the bacteria does this, they saw one more observation, that this.
Kritika, there's a request that you go into presentation mode. Is that possible?
Okay.
Thanks.
Better? Okay. So first observation of the scientists was that there was this extra chromosomal DNA. Second observation was that this extra chromosomal DNA was given to another organism So far, people had only known what is called vertical gene transfer, that is from the parent to the progeny, parent passing on their chromosome to the children. But then people started finding out that lateral gene transfer, where one organism will just give the genes to another, they started observing that this is also happening in nature. And then we have this natural selection or survival of the fittest. These rules actually allow anything new that comes in to actually start dominating, or it will start accepting anything that comes new, because the assumption is that anything new that happens would probably make the organism better fit, or would make the organism survive some other upcoming disaster or something like that. So nature itself has this process where it will select something that comes newly. These things were observed. Then came this process where you had conventional breeding techniques. So I think most of the organic farmers, or all of you, because you're already in the field, you would be knowing how conventional breeding is done. Here we wait for every generation and then see if we can modify it. So in conventional breeding, suppose you find a variety which has a bigger fruit, or any trait that was interesting, or you want that to continue. So ideally what they would have done is they would have they would have taken the pollen grains of that plant and back crossed it with the parent. So what will happen at every generation, because nature wants new things always, it would replace the original one completely. So by back crossing, the original copy of the gene would be replaced naturally. Here we are not meddling with the gene in any way. We are allowing the plant to decide that. In artificial methods, we have two varieties. We have cisgenesis and transgenesis. In cisgenesis, it is when family crops, or organisms which are related to each other. Like for example, potato and tomato are from Solanaceae. So if they are from a related family, if you can transfer genes between them, that would be cisgenesis. Transgenesis is when, like you have a gene in bacteria and then you transfer it to a plant, or a viral DNA into a human being. So these were also possible based on one observation, that lateral transfer is possible. So with that observation, they started attempting these. Say they took this extra chromosomal DNA from the bacteria, they added whatever gene they wanted to add, they tried transferring it to organisms, and they saw that it worked, because nature always selects. So for this transfer, whatever you use, that extra chromosomal piece of DNA in bacteria, so the name of that is a general name is a vector. Vector is another name for a vehicle. So we are just using these as vehicles to transfer genes of our interest into an organism. So we have various vectors for various situations. And this choice of vector depends on how much DNA we want to insert. So if you have a gene which is really small, that is of 0.1 to 10 kilobases. So one base is like one letter in that DNA that you saw. So when I say 10 kilobases, we mean 10,000 letters of DNA that you want to transfer. So with a plasmid, only that much is possible. But if you want to alter things in the plant chromosome or an animal chromosome, you would need something that would carry far more amount of DNA, and you use these kinds of vectors, yeast artificial chromosomes or bacterial artificial chromosomes, or you use viruses. So these are used to transfer genes. So this is your genetic engineering. So this was genetic engineering that people had been following for a very long time. This is not gene editing, this is genetic engineering, where you have a gene and you cut it and paste it inside that vector, package it in that vector, and then transfer it into a cell. So you can do it inside a bacteria, it'll just go in, and when the bacteria divides and multiplies, grows in number, it'll also multiply the gene that we have put in. It'll start making lots of copies of them, and all of that will be available. when you want to insert something into a plant genome, it wouldn't be done to a whole plant. So they would take the embryonic state. So when after the pollen grain has fused with the ovum, you may have the primordial seed or whatever. So there the gene is injected. So when you inject the gene, they would also, along with that, add some thing that they can use to detect the cell that has got it in. So suppose I put one gene of interest like this in orange circle, there is this purple thing that you've added. So that is the one that you wanted to add. Along with that, they would also add, say for an example, an antibiotic resistance gene or a metabolite gene, which means this particular organism initially would have been growing only in the presence of glucose. Now you add some gene which would enable it to grow in presence of galactose. So some such modification they would do so that only those cells which have got the gene would survive. So after injecting it, to make sure that they are only taking those cells which have got this gene inside, they have this selection procedure. So only those cells would be able to grow in that particular medium. Once they get those cells, they would take it and put it in a rooting medium and then shooting medium, and then they would get a small plant and then they would transfer it. This is how it is, genetic engineering would be done. So to sum up, gene modifications that can happen in an organism, you can generate these things by randomly mutating. So you want to improve crops, what they do, they would probably subject it to harmful radiations. Automatically some change would happen in the genome and the plant would become slightly different from what it was. Then crossbreeding with another variety. So now there are so many now. I saw some video where it was like pomegranate and lemon, they have crossbred and they have a new variety. Then you have something called polyploidy. So we saw that every chromosome there'll be two copies of it available. So instead of keeping those two copies, they would add like 5 or 6 copies more of a chromosome in which all the desirable genes are there. So that they would add more copies of it, which is polyploidy. So here they have also given examples of what was developed using each of these technologies. Then there was this uh, um, technology where it was protoplast fusion. So things in which you couldn't do a pollen transfer or a gene transfer, I mean before gene transfer, they tried if they could just fuse two cells together. So here you don't have a male plant, female flower, nothing like that. They take directly, normal cells. So these are not sex cells. These are normal cells which are then fused together to give a new variety. So this was another thing. And then came transgenic plants. So whatever we saw so far with genetic engineering, using that they started generating transgenics. Now we are having gene editing as a tool to again generate genetically modified crops. So this is just a comparison to show how much variation would happen if you use this technology. So the panel on top, if you see this picture, you can see a coin and the size of the cob in comparison. This is with the unaltered plant. Now this is when you have a genetically modified plant. Now this is the size of the coin and this is the size of the cob. You can make it this different from its parent. Whereas when you use traditional methods of breeding, you would have the one in the middle is your crossbreed between the parents on the sides. So these two combine to form this height, whereas in a genetically modified variety, with just one modification, you would be able to get something of this size. So we've seen genetic engineering so far. Now I'm just pausing to just let us assimilate all that we've seen so far. So I hope we've been able to understand that all living organisms have cells where DNA has codes for specific functions. And then bacteria have DNA that's not part of the genome, and these were used to introduce DNA into cells and they were called vectors. Then we saw that even viruses can be used. When you have a big chunk of DNA that you want to transfer, you could use other vectors. Once introduced, the gene has to go through transcription, translation, and create modifications in that cell, and in turn it would produce something in the organism. So now let us try and understand gene editing. So again, here I want to pause and interact. Please use the chat box and tell me what are the steps for a document editing process. Suppose I'm the author of a book and I give you the book to edit. What all will you usually look for as an editor? Okay, typos. So I would put that as spelling corrections. Okay, spelling, spell check, grammar, replace. Okay, look for missing words. So I'll put all that together as replacements. Okay, content. Editing in terms of punctuations. And gaps. So suppose there was okay, so I think we've got pretty much whatever we wanted to understand gene editing now. So now these are the steps when you have to edit a document, right? Exactly the same thing happens in genome editing as well. So if you assume that the genome is a book with a lot of text, so each of those sentences are your genes. So you feel that there is one gene which you don't like, you want to modify. What you would do, if there is a mistake, you would change the spelling. Then you would change the grammar of that sentence. Then you would look for a thesaurus, replace that word with something else if you feel something is repetitive. Then you would check the content. If you feel that paragraph doesn't make sense, you would remove the whole paragraph, replace it. Then what you would do is, suppose there was an extra gap, you would use the backspace button. If there was no gap, then you would use the space bar. These are the basic things that you would do. So now look at the same things in terms of genetics. So this is genetic mutation. So suppose I have a different spelling. So this is like a spell check. Suppose I had my original sentence was I'll be by the lake. When I say point mutation, where I just change one letter, I'll be by THW lake. That was the error now. Point mutation 2, instead of lake, I'm adding cake. Okay. Then you have nonsense mutation, where something got randomly deleted and it doesn't make sense anymore. Then you have something called frame shift. So suppose in this sentence, I start from LLBE, then it wouldn't make sense again. So these are the types of mutations that are there, that are already there, and these are the things that you can add. So here, by just replacing A, or by deleting A, the cat and the dog, this sentence would become the AT and the dog, which wouldn't make sense. Are big. So the cat and the dog are big. The at and the dog are big. So by deletion, this is the kind of change that would happen. This is very much the change that happens in gene editing. Now, when we say we identify an error in the document, what we would do is we would remove the word and then insert something else. In genes, how do we remove this? So for that, we have a group of enzymes called nucleases. So if you recall, we called DNA as deoxyribonucleic acid. So when I say nuclease, it's an enzyme or a protein that would chop DNA. So we have something called restriction endonuclease. These are those that are present inside the cell which will restrict, which will cut the DNA. So we have lots of varieties of these cuts possible. You could cut, both the strands of two. Suppose I have two strands of DNA, I can cut making equal, things like this. And then I can have one cutting where I can make something like this. Okay, so here I have cut a little bit from this side. Here I've made a staggered cut. So there I in the third instance, when you see it's cutting here and here, so it is making what are called this kind of ends. Okay, these were the ones that have been used in genetic engineering. Now we will see in gene editing how these have been modified. So these are proteins which can just go around the DNA, identify a particular place where it can go and chop. So with respect to gene editing, when you look at Cas9, CRISPR Cas9, the Cas9 is the protein which is going to act as this nuclease, which will go and cut the DNA. And then in this technology, which is TALEN, which is called transcription activator like effector nuclease. So that is another technology which involves a nuclease which does cuts like this. Then you had zinc finger nuclease based gene editing. Again there, that the nuclease has a zinc finger motif which will go and cut it. So these are the nucleases they use to edit. So we saw what are these changes that we can add to a gene. What are these mutations that we can incorporate in a gene. Now, while incorporating these mutations, how do we make them incorporated in that particular DNA? So we remove the original version that is there in the DNA and add something else. So for this removal, these nucleases are used. Now coming to, okay, now let us think of this situation. When something happens inside a cell, like when somebody comes and pulls our hand or hits us, will we be quiet? We won't. Sometimes we ourselves cut our hand. We'll immediately go and try and repair. So all cells have basic repair mechanisms also in their own system. Now this also we have to understand so that we understand the technology better. So what are the kinds of repair that would happen? So here we have base excision repair. So in the DNA, if you saw, there were two strands. So one of them had, suppose one of them had A, the other one would have the complementary alphabet T. If one of them had G, here you'll have C, and so on. So if there is a change in only one of the strands, that is called a base excision. So when there is a base excision, only one base is lost. There is a kind of repair which involves all these enzymes to fix that. Suppose I have a nucleotide excision, then I have both the alphabets removed, like we saw in the thing that doesn't make sense. So there I would have another repair mechanism that would be stimulated. Then another repair that happens is homologous recombination. This again is nature's, nature does it on its own. You might have heard, sister chromatids having homology and things like that. So what do we mean by homology? We'll just try and understand, because this is what is involved in gene editing. So suppose I have a trait or a character called height. So you can express height in terms of shortness and tallness. So each of these traits, you would have two versions of it. So those two versions, how they balance out each other would be how the organism would express it. So if I am a particular height, it would be the base, the combination of these genes from my parents that would have caused this. Suppose something happens in one of the genes at a particular gene which involves a particular trait, then nature would use the other copy to repair it. right? So suppose I had a big assignment I was writing. Somebody poured water on that particular page. With reference to context, we would be able to understand what that sentence would have had as a word there, right? Or somebody presses the backspace button by mistake, some point, points from your document get deleted. You would compare it with the context and you would add something there. That is homologous recombination. So for that, you have a big machinery which does that. And then you have non-homologous end joining. So in this, without even looking at the homology, it's like a quick fix. So when there is a cut, what they would do is, suppose you look at carpenters repairing some, you have a piece of wood that has been broken like that with lot of splinters. They would first smoothen it out, right? And then only fix it. So this non-homologous end joining would remove the splinter. So it will remove extra DNA from there and then join. So these are the editings that happen.
Sorry,
Yes.
Kritika, have you missed the video on the earlier slide?
Should I go through that endonuclease video? You think
Yeah,
they
I
would
think so.
like to see it? Okay.
Yes.
Okay. I didn't know whether they would want to see, so I just skipped that. Okay, I'll just show you that endonuclease video. So what I had was a video of an endonuclease. It's just an animated video just to show how this works. Just a second, maybe I'll.
You have shared the screen, no?
Not yet. Uh, Yeah. So this is a video which would show how endonuclease would go and cut. So this is this enzyme which would scan the whole DNA, find a place where it can cut. So we already know what a nuclease is now, so we'll see how it works. So suppose this is the piece of DNA that we have to cut. This nuclease would go through it. So this blue thing that is coming there is the nuclease. It's an animated version. So it'll just go spin around the whole DNA, look for a specific place, and it'll go and cut. The repair thing would come and paste something there and repair it. So I didn't want to give too many names for those. So okay, just for whoever would be interested, these enzymes are, one of them was an endonuclease, the other one that attaches or pastes is called the ligase. So we saw what all kinds of repairs are possible, and in gene editing, the kinds of repair that people have been exploiting are the homologous recombination and non-homologous end joining, these two kinds of repair. So in homologous recombination, just to remind, you have a piece of DNA where you want to introduce something. So you introduce that information in that enzyme so that it will go and specifically look at that place and cut there. Then once it is cut, the organism would be looking for how to repair this. You also provide a little bit of DNA which has homology to parts of it. So the cell would want to immediately fix this problem and it will attach it there. So by attaching it to the based on some parts of it which had homology, you are also they've started introducing pieces of DNA which they want to put in between those places where there was homology. That is homologous recombination. Once that is done, this non-homologous ends are joined so that splinters are removed and then joining happens. So each of this is used for a different kind of editing. Now this is one other aspect people have been exploiting. When you look at these repair mechanisms inside an organism, there are two types of repair. So we call it SOS repair. So this again, I would tell another thing that we can relate to. Suppose you had a plumbing issue at home. There were two plumbers whose contact you had. One fellow would immediately come and fix it. The other fellow would probably take time, but then if he does it, he would do such a perfect job. So whom you would call would be based on how what is your current requirement. So suppose you have a big damage, you want to first immediately fix it and then worry about going and making things better later. So based on situation, the cell will decide. This is another thing that genetic gene editing people have started exploiting. So they would they would have an enzyme which has been modified in such a way that it will be available as that always available person. It will go and fix it. Once that fixing is done, you have actually introduced your gene of interest. So this is the thing that people are exploiting. So just to reiterate all the modifications about gene editing and let us understand. Suppose you have insertions, deletions. So insertions in the sense, suppose there was that extra word that you didn't want to add. Sometimes when we type also, we would have probably by mistake added two A's or two the's and things like that. So those are the insertions. Suppose you missed writing a word, that was deletion. And then we discussed about base excision, nucleotide excision. So one single letter going off, two letters paired to each other going off. Then there is a type of thing called mismatch. So suppose for one letter, something else was on the other strand, that was mismatch. Now then the other one is double strand breaks. This is the thing that people are using for these technologies, that is CRISPR and TALEN and ZFN, zinc finger nuclease. For all that, this double strand breaks and the repair for that particular situation is your non-homologous end joining and homologous recombination. These are the fixes for these. This group of things are the ones that are used extensively in any of these gene editing tools. So just to stop a little bit and analyze, we say if you just think about that slide where we spoke about frame shift. I was thinking of this dominoes effect, or some of them call it the philosophical way of thinking. It was that butterfly effect. So suppose this domino, I mean, nobody hit it, the rest of the reactions wouldn't happen. Or I would also have another example to understand the situation where in a blind person's house, he would have actually set the house in so well that they would exactly know how many steps would lead them to the door, how many steps would lead them to a chair, and so on. Suppose there was a stranger in that room and they had rearranged the furniture, what would happen? This is actually what we are doing while we do gene editing. And that is why, as a biotechnologist, I feel very bad about it, because when we alter one gene, that alteration wouldn't stop with that one gene, is what I feel. Because for one gene, it's not that gene's expression alone that is affected. All that is before that, all that is after that, also get meddled up with. So this is why we should actually be fighting against gene editing, because for us, when in genetic engineering and gene editing, what is the major difference? In genetic engineering, a whole gene is being replaced, which is again wrong. But here they claim that here we are only altering one mutation. We are only changing one nucleotide. So somebody is raising the hand. Should I interact?
Please put something in the chat box. We
Yeah.
can't entertain raising the hand. Sorry.
Yeah.
Yeah. You can use chat
Yeah.
box.
See, I have not even gone into the technology, so that is why we might not be able to interact right now. So this is why I feel that we should be opposing gene editing as well. We should consider that also as a kind of genetic modification, because we can't say it's just one word that I have changed. Suppose you had a holy book in which you have introduced lines which don't make sense at all. It's actually, then you can't say, oh, I only put 4 lines which don't make sense. That wouldn't be so right. Suppose you had a whole book about how yeah, so this is the thing. Suppose you had a holy book, you can't write nonsense in just one paragraph. I have altered, I've just added something that I wanted. It's not a right moral thing that anybody should be doing. So that is the thing. So now we are getting into the technology per se. So what is gene editing? The most famous ones are these. You have something called meganuclease, you have zinc finger nuclease, you have transcription activator like, that's the expansion for that, transcription activator like effector nuclease. ZFN is zinc finger nuclease. CRISPR is a bigger one, so that stands for clustered regularly interspaced palindromic repeat, that is CRISPR. Cas9 is a nuclease. So CRISPR associated, um, associated, that is Cas. CRISPR associated nuclease, Cas nuclease. So the Cas9 is a modified Cas nuclease where they have made sure that the original reason for which the organism had Cas9 has been modified. They've changed it in such a way that they can use it now for gene editing purposes. So here I also have a small inset where I'm just telling you about the speed at which this is happening. So they first reported or just looked at this, they discovered that there is something called CRISPR. There is a associated nuclease called Cas. These were, just observed in 1987, and nothing happened till 2002, thankfully. Once this CRISPR was named, from 2002 to 2020, so much has happened, right? Within 18 years, so much has happened. So many genetic modifications have happened. Even a human twin, human twins have been generated through gene editing in China. This would be a shocker. I would just show you pictures of those twins also. So this is going to actually raise a lot of moral questions in our minds. So these 3 people have been credited for the development of the technology of CRISPR. I'm not elaborating about Talon and ZFN, because CRISPR is what is supposed to be the most efficient tool, most cheap one, which can be done in any lab. For Talon as well as ZFN, you need a little more equipped lab for carrying out these. But CRISPR, they claim, can be done in a kitchen also. So there are kits for CRISPR available that even lay people can purchase. So even if a high schooler is a little over enthusiastic, has learned a little bit of genetics through online courses, they can also purchase the kit for CRISPR. So I thought I would talk more about CRISPR. The difference between these two, just the 3 of them, just to that I would share. The difference is only in the nuclease, if you see. So we saw what a nuclease is. It goes and cuts. So in CRISPR-Cas9, we would be seeing that. Here, this Talon, we saw what is transcription in the first slide, right? So transcription is the transfer of information in the DNA to RNA language. So I have the original copy in the DNA. I just, the transcription activator would begin the process of It is a protein which will go and attach to the DNA, and it would call another protein to come and attach below the DNA and start making a copy of the mRNA. So that is a transcription activator. So this one is a transcription activator like effector. So this would go and bind to the DNA and it will cut. It will bind just like a transcription activator would bind. And it is a nuclease, it would cut at a specific place. How transcription activator would bind would be based on a particular sequence that it can identify in the DNA. There it will go and bind. This nuclease needs two things to be bound to two sides of the DNA. So this technology has now been overthrown by CRISPR. Zinc finger nuclease, all these nucleases would have various, domains or, places. So the shape of it would be like this, and suppose I have the DNA, how I can hold it? Can I hold it like this? Can I hold it like this? So these shapes and what is present at that shape. So this one has a zinc finger motif. So it uses that particular shape to go and cut the DNA. This is the basic difference between these three technologies. About CRISPR, we would try and understand what's CRISPR, because that is the one that is really dangerous with the way it is now available. So there are two videos. One is about how CRISPR originally started. I mean, how did they discover CRISPR? The second video is about how they are currently using it. So both are 5 minutes or 6 minute videos. I hope it's okay. Again, I'm not using I would be muting the videos original audio and I would be speaking along with it, like, I'm explaining the video. So I'll stop sharing this. I hope, it's clear and I'm not going too fast.
Perfectly all right.
Okay. Yeah.
Yes, we can see.
Yeah, the video has to start playing. That's it.
Hmm.
Yeah. So this is actually about how they discovered this thing called CRISPR. Just like an introduction. So everybody is looking at what is going to be the next big thing. And everybody is looking for that magical discovery or eureka moment as scientists. So that is how these people also began with the CRISPR technology. So we are going to understand how they discovered CRISPR. So initially there was this scientist who discovered that in bacteria, especially in E. coli, there were stretches of DNA which were palindromic. So I hope all of us are aware of what is palindrome, like Malayalam from here to there, from this side to that side, it reads the same. So here the sequence is ATCTA, and from the other side also it is ATCTA, if you see. ATCTA, ATCTA. So you had repeats, palindromic repeats. So here was a palindrome, then was a spacer, then another palindrome and another spacer. So this is what is a palindromic repeat. And each of these palindromes, if you see, were approximately 30 bases long, not very, very small ones. To a stretch of 30 bases, it was a palindrome. And they were not able to understand why it is like that. I mean, or its function. Then in 1990, another scientist found that not only in bacteria, but also in archaea bacteria, in those also which are supposed to be evolutionarily before bacteria, in them or in these organisms also, these kinds of repeats were found. So then when something is found between unrelated species, then people look for, I mean, they feel that there must be some importance for this palindromic repeat. So they felt it must be important. So they went and, so then they named it CRISPR, clustered, regularly interspersed, short palindromic repeats. Then they took out those spaces. The palindromes, everywhere it is the same sequence, but the ones that are between them, the spaces, they took those spaces and then they did a bioinformatics search. Even now there are these tools called BLAST. So there is a website, if you just go type Google, BLAST in the Google, it will take you to a database of all sequenced genomes, genes, everything. So if you have a copy of the gene, even if you want, you can just go type COVID-19. It will give you the list of the whole genome of COVID-19. Then you can go compare any of the genes that you want to compare with using this particular tool called BLAST. So the expansion of this is basic local alignment search tool. So it will take your gene, compare it with that, wherever locally it is able to align with that original copy, it will align and tell where all it is matching. So when they did this alignment, they found that this sequence was from a virus. So all the spaces were viral sequences from the virus. Now just let us, I'll just tell one more thing. For us, we have pathogens that infect us. So bacteria will infect us, viruses will infect us, fungi infect us. For bacteria, viruses infect bacteria. So for bacteria, viruses are their pathogens. So with this information, let us see this again. Go forward. So they were worried about why does a bacteria have a viral sequence in it, and why is it conserved, that is along along species which are completely unrelated, like the archaebacteria and bacteria. How can something that are unrelated have sequences of viruses in them? So this was the next point of concern for people. Then they went ahead in a yogurt factory. So in yogurt, all of us know that we curdle milk with bacteria, right? So they found that this bacteria was not a good culture. So the yogurt that was formed was not at all good. And then they found that that bacteria was infected with a virus. So this was found by Philip Horvath. So then he found that there was these other kinds of bacteria which had those spaces. Those were able to successfully fight that virus. So this was the first time that they found that these spacers were probably like the immune system that we have. So when we have an infection coming inside, our body produces antibodies against that infection. So this was the antibody equivalent for a bacteria. So if that bacteria had that particular sequence, it would be able to fight that virus. How does it fight? Would be what is CRISPR technology. So once it finds, so it, this is your Cas9. So what happens is the bacteria has this sequence which has palindromes and spacers. Now the spacers are the pieces of DNA that correspond to a virus. So what the Cas9 protein had actually been found to do was it would go through the whole genome of, okay, so it will take the spacer and then make a RNA for that and keep it ready. Whenever there is some extra DNA floating around in the cytoplasm, it will go and check, okay, check whether it is a virus DNA which corresponds to that spacer piece. Then it will immediately cut the virus's DNA. I hope you're able to imagine that. So it's like the antibodies. So what do antibodies do? They will be floating around in the body. Whenever there is a foreign protein, it will go touch it and see, okay, is this our protein or something else? Then it will go tell the body to produce more of antibodies. It will go trap the pathogen and throw it. Just like that, in a bacteria, it would go through the whole virus genome, check if this piece of DNA belongs to the virus or the bacteria. Once it is able to identify that it has a spacer equivalent, it will immediately chop the virus's DNA. So that was done by the Cas9 nuclease. This was found by John van der Woest. So he confirmed that by altering the spacers sequence. Whenever he altered the spacer sequence, he found that the bacteria was not able to recognize this virus. So he then confirmed that it was like an artificial flu shot for a bacteria. So if you alter this, then you could do it. So this person, Emanuela Charpentier, she is one of those scientists you saw in that picture. So what she did was, they found that there is an RNA which is that equivalent of that spacer that is part of that protein. Okay, now again, it might go out, bounce off your head, just to let us understand this a little bit. We have this DNA in the bacteria, which has this spacer. This spacer has the information that tells that has a information which is a virus DNA's information. You have this protein which would make an RNA which will have the sequence and keep it ready. The moment there is a virus, this RNA would go and scan the whole genome of the virus. Wherever this RNA matches, the protein would go chop it. This is what is happening. So the protein would not have the genome's tracer DNA. It would have an RNA copy of this particular spacer. So now what Emanuela Charpentier did was she found that there is an RNA also in the nuclease, which is the one that is recognizing this, virus genomes. That is the one that is going around and checking whether this thing is similar to the spacer or not. So in 2013, what she did was that they started modifying that RNA now. So you can actually tinker it. How? So let us think of the situation now. Now I know that there is, I have a tool with me which will compare with a DNA, and wherever I find that this DNA is matching whatever I have as a copy, this protein is going to bind there and chop at that place. So this was exploited. How did they do? This RNA that is there in the protein, if you change the sequence of this to match a specific place in the whole genome, then this protein will go bind at that portion in the genome and chop there based on this RNA sequence. So they called it the guide RNA, and they started changing the sequence of the RNA. And then they started doing it initially with test tubes and petri dishes. And Jennifer Doudna, that other scientist that you saw on the picture. So these three people modified the RNA. They added extra pieces that they want to introduce, and then started using it as a tool to go and chop DNA or chromosome at specific places that they wanted to add something to. So now with this technology, what they have been able to do is, this lets you take your cutting enzyme to a particular place in any DNA, in any. So it can be initially it was actually present for virus DNA, as a defense against virus DNA. But now they have started modifying the protein in such a way that it would have a RNA piece. Okay, that RNA piece would take you to any gene on any chromosome anywhere. So it can go and do a targeted cut. Now there is another video. I hope it's okay. These things, when there is a pictorial representation, it is easy for us to assimilate. So I always have videos that will help us picturize the whole thing. Now we know that this protein has this modified RNA. Okay, from there the story continues. So now this is again the nucleus that you saw in the first video. So you have the nucleus. We are going to get into the nucleus. This is the chromosome. So chromosome, we saw already that it has this DNA which is packed like this. So now let us look at the DNA. This is that Cas9 protein. So this is having the piece of RNA that we understood from the previous video. And you have this RNA, which you call it as single guide RNA. So it is a single stranded RNA, which is going to guide us to the place where it will match with the DNA. So now it has gone and bound. So it will go look for a place. While it looks for the place, it will look for the sequence called PAM sequence. Now PAM stands for protospacer adjacent motif. So we know that there is a spacer, right? So proto means primitive, or just before. Just before the spacer, you have a adjacent. So the motif that is adjacent to the spacer is your PAM sequence. It will go and look for that PAM sequence and go and bind there. And then it will uncoil the DNA, uncoil it in such a way that this particular sequence aligns with that RNA, the original, that RNA that the protein had in it. Then it will go and chop exactly at a particular place. Once it is chopped, the repair processes that we saw before, they would go and repair that place. So suppose this repair enzyme is one that is the quick fix one, it will shut off the whole gene, which is beyond it. Then what people started doing was they altered the protein in such a way that it will go and make specific changes. So they had these additional enzymes added to that protein, which will go and do one specific modification in one nucleotide alone. See, it will just go and modify that nucleotide alone. So cytosine would get mutated into thymine, in which case you would be doing a spell check correction kind of a thing. So only one thing you would modify. By modifying, what will happen is that place which would have originally meant cat would start now meaning bat, for example. So it would either bring a different amino acid at that protein's place, or it would do something like stopping the gene from expressing itself. So the shape of the protein they kept altering so that they could meddle with what all functions the protein would do. So they added some other proteins to the Cas9 protein so that when it sits on the DNA would stop functioning. Like here, if you see, they added extra proteins which will then hold the DNA, stop it from, or in this case, it started making another. So from the later part of the gene, it should start making some other protein, or it would stop the DNA from functioning. Okay, block that gene, for example. So this is what is called gene silencing. Or you can also use it as a fancy thing to go look at how where a particular gene is located in the chromosome by attaching a fluorescent protein to the Cas9 protein. So by that you can in fact color the chromosome, make it go and attach specifically to some genes and so on. So these have been the modifications people have been using Cas9, CRISPR-Cas9 for. So here we saw three kinds of modifications they have been able to effectively do with CRISPR-Cas9. So one of it was, deletion. You could delete pieces of the DNA, then you could modify. So you could do a spell correction, spell check correction, or spelling change, or you could completely cut pieces of it, remove it, add something else. So when you add, they would add anything else along with it that the cell will start accepting it. So, and then you can also do gene silencing. Now, if you look at why these people are promoting this technology, all of them speak about specific diseases where there is only one mutation. That one mutation was causing things like sickle cell anemia and diseases like that. There is only one mutation. If you can go to that genome and correct that one gene, that person will survive. There are these conditions like muscular dystrophy and things like that where it is only one gene error, or one mutation, one single point mutation, where by correcting it that person would survive. For those things, these people are promoting this technology. As a scientist, I also feel happy that whenever a technology is going to help somebody, we are always going to welcome it. We are not these cave people who would say, no, I don't want to harm nature, I don't want to get out of my cave. That is not the kind of thing that we are looking at. But if it is going to harm generations later, then we shouldn't do it. If it would help one person, let them do it with that one person, go ahead with it. But by doing these modifications, if we are going to corrupt the whole genome, generations later, then that would be harmful. So this is why they are promoting CRISPR technology, because it would be specifically able to modify one particular gene, one particular place at a time. But we just saw about this error correction enzymes, right? So whenever there is a mutation, the proteins from the cell would go and do repairs. So I was telling you about these SOS repairs and the other repair. So we had two plumbers who would fix it fast and do it. So any of these damages done by Cas9 would actually call in for SOS repair proteins. So because SOS repair starts functioning, sometimes, in fact, most of the times, some damage happens to the DNA. So here, from here we are beginning into the concern parts of it. We saw how the technology works. So to an extent, maybe we would be thinking, oh, maybe it's not such a bad technology. But then let us look at this now. So many organisms have been edited already using ZFN, TALEN, and CRISPR in invertebrates, in plants, in vertebrates. So many organisms have been edited already. Okay, so these are some of the traits for which they edit. If you see, myostatin is a muscle protein. If you see, some of these proteins, in fact, some of these organisms, no one knows what the function of the gene is. And these people, just for curiosity's sake, they turn on the gene. Why would somebody want to do that? Right? If nature chooses to silence it, we shouldn't be unnecessarily opening it up. It'll open a Pandora's box, is what I feel too. So some of these pictures are from another scientist's presentation, and he had happily shared it for us to use it for any kind of purpose. So in fungus, they engineered using CRISPR technology, one enzyme which was producing brown color on the mushroom. That gene was silenced. So you have mushrooms which would not look brown at all. So it be visually appealing, it seems. But then what if that modification would trigger some other pathway? No one studied that. Then large number of gene edited crops which have not been approved by many of these regulatory authorities have been let out actually. Then they had gene edited farm animals like the hornless cattle. I mean, having a hornless cattle, I don't know what benefit it has for the human beings. It wouldn't poke you or something. For that purpose, should we be meddling with the chromosome of the cow itself? These are things that are of ethical concern. Then they have, these kinds of things where they have extra muscly pigs for, extra meat production. So here I have this human genome editing, which is again a possibility of this particular technology. Twins, Lulu and Nana. You can see the picture, I hope, of those two children. They have been done. So in China, these twins have been gene edited. So if we look at regulations on gene editing on organisms, I have just added these links. These links would take us to those specific papers which talk about the whole scenario throughout the world. The ones that are the darkest have the tightest regulations. In all other places, you have review of existing process and regulatory systems have just been begun. I mean, they've just begun. So here also in India also, the Ministry of Science and Technology has started a Department of Biotechnology. Under that, they have biosafety guidelines for genetic modifications, which is just in a primitive primordial stage. Nothing has been laid down as guidelines so far. They only have collected everything and put it there. So our concern is, will it apply for labs that have already got affiliations from the ministry before? Are they going to have some survey committees which would just go do random checks in these to see if the regulations are followed? I used to work in a lab which I don't want to name. We did not have any regulations followed in the lab. We had just, see, I was in a crystallography field. It was attached to the physics department. Some of us were doing biochemistry, which means there were no facilities to do these kinds of research in our lab, which means anything that comes out of it would be just dumped down the sink or in the dustbins without any precaution. Now with COVID-19, each of us has now started using sanitizers to just go out to the grocery store. But in a lab, I have had students who would handle typhoid organism with bare hands. I have seen with my own eyes. So these are things that concern me actually. And how effective is this technology? So we saw these these would trigger SOS repair. So I have a collage of things that you yourself can Google up and see. If you just Google this particular line, CRISPR-Cas off-target cleavage, will it always go cut at that point, or will it go randomly cut somewhere else? Okay, this was not checked, for your kind information. If you go check there are about 18,000 papers which say that it will go cut anywhere else also. It is not always on target. So what do we mean off target and on target? We saw that the Cas9 would go and bind to the DNA and cut at a place. It might not always cut at that place, it seems. It will go cut anywhere else also, it seems. So this is again this blind man's house rearranged kind of a situation, right? If we meddle with nature without knowing what would happen as a repercussion, it would lead to dangerous outcomes. And if you see, this is again one thing. In the first slide, I told you about the sentence formatting, right? We have a jumbled sentence. So the mRNA, before getting out of the nucleus, I said would have some pieces of it removed. So in plant genomes as well as animal genomes, whatever is removed is called an intron. So far people had been thinking that does not mean, that is just junk DNA that can be removed. But there are so many reports which say that the introns are very, very important. They are the ones those are the steps that the blind man would count. Okay, it will say, okay, 10 bases later is where I have to chop. And then after that, 15 bases later is where I have to chop again and join. That is the introns purpose, because sometimes this chopping might not be perfect. So the nature has given those introns so that if we stumble upon another one also, you would probably get to the right place to cut. So these introns, which were thought of as junk DNA, were the positions that were used to insert genes during gene editing because they thought it is junk. But if you look at some of these papers, if you delete the intron, it seems the whole gene would get shut. If you delete an intron, it would start doing a frame shift where it will start reading from two, three things before and make a meaningless protein. So these have also been reported. So where you insert the gene, how is it inserted? All these actually matter and people are ignoring all this. Some more potential concerns. So when you knock out a gene, sometimes it is not completely knocked out. What is knockout? Suppose I completely shut off a gene's expression. Sometimes that shut off is not complete because you saw in that video, it'll go bind to that DNA and just, tweak it like that. Sometimes it'll be loosely fit, it'll come off and the gene will start expressing. So sometimes knockout is not complete knockout. Then when we do with human embryos, something else happens. Who was going to regulate all that? Then when we say insertion of an editing template is, so each of these papers talks about what all can go wrong. So what do we need to do is that we have to definitely have regulations in place all over the world. So if we say genetic engineering is wrong, gene editing is also wrong. If we say genetic engineering is wrong, gene editing is wrong, any product that comes out of it should also be banned, right? So we saw that lateral gene transfer happens, which means genes can be transferred from one organism to another. Okay, fine. True that we have been eating vegetables for so long, we don't develop green color in our skin. Fine. So gene transfer hasn't happened. But how sure are we? Because how did we get viruses? How did we get so many varieties of viruses suddenly? It might not get incorporated into us. What if it floats around, causes a harm as a new pathogen? So these are the reasons why even gene edited products, genetically modified crop products should be coming under some regulation. So I think we've come to the end of this session. Just to summarize, so through this thing, we've understood what a gene is, how is it naturally transferred across generations, how can genes be transferred to unrelated species both naturally and artificially. We saw what is gene editing, we saw what was CRISPR-Cas technology, and now the questions in our mind as we go, go home after this, be at home after this should be, is it really foolproof as a technology? What should we be doing about ethical considerations with regard to indiscriminate use of gene editing? So thank you for your attention. I just want to wind up with a cartoon which says, okay, once this person had taken the child for curing allergies using gene editing, and then they wanted to modify everything else about the child. And are we actually thinking of the ethical aspects of it? Yeah, I would want to thank you for your attention. I hope it was useful.
Thank you so much.
Yeah.
Thank you so much. It was a really a good undergraduate class, and we definitely like to be a student, whatever the age. There were scientists, old age people, senior activists, farmers, anything, anyone. It's okay. But you selected good slides, videos, all that, and in 90 minutes now we are definitely more confident to know and also more eager to know. Of course, more eager to know this. You can see the questions.
Yeah, I
Would you
just
like to?
opened the chat actually.
Yes, yes. And yeah, someone has.
Asked about
Uh,
the case we filed. Yeah, I'll talk about
So,
it. And yes.
what I have done, Kritika, is
Yeah,
to
I saw.
keep adding all the questions again and again as they were coming.
Hmm.
So, for now, we have some 14, 15 questions
Yeah.
towards the
So
very end. May I request
yes,
others
I have
not
seen
to
it.
start
If
chatting?
nature, so I saw this question about if nature is able to repair the genes, how do mutations happen and get propagated? Yes, so there is a repair process. So when they talk about this repair, that happens. That is why they, when in the lab, they always attach a marker along with it, so that the plant cell or something is forced to keep on repeating it. That's why if you see many of the genetically modified crops like uh, this BT and things like that, after the first generation, the crops didn't survive, because after the first generation, the whole thing was replaced. So nature would do the replacement, of course, eventually, not always. Is this a new piece of DNA from different species? Um, by that, are we trying to talk about things that we use to insert? Yes, of course. Anything that we want to introduce into something, like this golden rice. Uh, you're introducing carotenoids into rice plant, assuming that that would give us vitamin A replacement, which okay, you've replaced it, but you've also shifted a lot of genes which were next to it. Would it have an impact? We have to wait and see. So that is what the actual ecological environmental implications of these kinds of introductions or modifications would start showing up, and then it will be very dangerous. Is what are your views on using technology to repair bad gene of an embryo? That's what. See, if it is going to help somebody, if one small point mutation can be corrected at the embryo level itself, then I think it's okay. But again, we don't know whether some of these enzymes are foolproof, whether this modification would stay as such that the after modification, will the cell correct it and make it bad again? These are things that nobody bothers about. CRISPR is allowed globally, yes. Like I mentioned, you can actually purchase those kits. So there is somebody called a biohacker. So he sells kits. And then what are the laws that govern gene editing? For now, European Union, and so I shared some of the links of the regulatory guidelines that were framed in some of the international meetings. So Argentina has the strictest laws, it seems, and they hosted a conference, and a lot of decisions were taken. So that picture of the whole world's countries that had participated there and are in the process of regulating were there in that picture. The link of those are attached. In theory, it is not possible to use CRISPR to build immunity from corona. There are people who are working on it actually. How is CRISPR different from GE or GM crops? So genetically engineered, genetically modified. So genetic engineering is when you are tinkering with already existing genes, by modify. So you are changing the sequence itself, like a spell check correction or a thesaurus correction. And genetic modification is when you replace it. The whole gene is replaced, modified, so altered. That is, so this whole engineering process requires genetic modification. For what kind of diseases have they experimented with gene editing? Um, some of them that I saw was muscular dystrophy, and then sickle cell anemia, kind of wherever there is a single mutation causing a disease, or in all those places they have attempted. Is gene editing allowed in China? In fact, this person has given a speech, I mean, in a conference, but then it seems after that he was taken into task, and Chinese government was not very happy about it. But I don't know why some of the Chinese labs still have gene editing work going on in their labs. It's just not healthy, an environmental issue, more ethical issue. Yes, it is an ethical issue. Um, Gene is not introduced but modified eventually. Pleiotropic effect will be there. Yes. Startup is using gene editing to make fragrance additives for food. What do you think about them? Many of these fragrance additives, anything that involves some of these, like these polyphenols that were deleted from the mushroom, most of these would involve either the fat metabolic pathway or some of these biochemical pathways, which are actually extremely complicated, and they have a very complicated feedback system. So it's like the valves in a big pipeline. So if I close this valve, the whole thing would get affected. So some of them have complicated feedback mechanisms, and we don't know what is going to trigger what. When we say feedback, suppose I have insulin, I take insulin, insulin would go and immediately shut the glucose production. So what will happen? When there is low glucose, some other pathway would start replacing it. Like that, by altering one thing, you shouldn't be triggering something else, like a domino effect.
I'm just sending you the next set of
Next set.
questions.
Okay.
And I think the question around what exactly is the difference between genetic modification as we knew it in BT cotton, BT brinjal, and so on, and this gene editing, a second question has been asked on
Yeah.
that.
So
Maybe you
that,
could.
yeah, that's what. So in BT, if I take specifically BT, BT had the introduction of a particular endotoxin, Bacillus thuringiensis. So Bacillus thuringiensis produced a protein which was detrimental to an insect. So what it was that particular beta crystalline proteins gene alone they introduced into cotton. So what happened was it was producing beta crystalline along with the in the cotton plant. When the insect came in by bit on the leaf, it ingested beta crystalline and the insect died. This was the genetic modification that was done with BT. So in that, you have completely introduced a new gene into a plant cell. In gene editing, what they do is specific places are identified and modified. So in a site specific fashion. So if I say paragraph 2, word number 3, I'm going and modifying. So it's that specific. That is the only difference.
Okay, so there's a question about in nature, when mutations happen, how long does it take for it to stabilize?
Uh, as the natural selection theory suggests, usually nature will take up anything that is added newly, because it will assume that it's going to help the organism experience some upcoming bad event or something. So nature always assumes like that. So it will look for 2 generations. So first generation, second generation. After that, if it finds that this change is not giving any benefit to the organism, it will delete it. That is why in traditional breedings also they would do it with 2 generations and then only continue with the breeding of that crop.
What are the advances in genome editing testing in detecting?
Whether it has been edited or not, is it?
Yes.
So ideally they would do a blast this thing. I mean, nowadays it's very easy. If you take organism, take out the DNA of it, do a sequencing in any lab. So sequencing can be done with just 4,000, 5,000 rupees. Whole thing they would sequence and tell you. Once you get the sequence, anybody can go Google it up, say blast, and then you would be able to know whether everything is intact.
There's a question on whether a gene edited organism can have foreign genetic material introduced.
Yes, they can. So when they do that homologous recombination, so it will identify those regions. It will only usually look at ends which are homologous. So, and then in the middle, they will introduce whatever they want to.
Mm-hmm. You mentioned in the beginning, you corrected me saying that it is reversible. What exactly did you mean by that?
Yeah, so this is what we said. Suppose after 2 generations, nature doesn't think this mutation works. It would reverse it. It would just, take it back to normal. It would repair it.
Mm-hmm. There's a question on biofortification. There's so many foods and crops that are being fortified these days. What do you think about fortification, and what are the negative or adverse implications of fortification?
For me, the thing is, anything unnatural, we wouldn't know the implications immediately. When we know it, we should be hopefully equipped enough to handle. So why unnecessarily fortify when like even this golden rice, that's what I keep saying. Why fortify it with the vitamins when you can take it as a separate supplement actually? So milk you fortify with vitamin D, vitamin A. Instead of that, why can't they take another food item that has it? So I don't know.
Okay. Uh,
So that is why I'm saying ministry has to have some way in which they would do these checks on not only labs that are asking them for affiliation or for funding, but to every private lab or anything that manufactures these kits. They shouldn't be made available to anybody who wants to buy it. So not only are the products and the procedures that have to come under the regulation, also these private labs which probably don't ask them for a funding, for example. So any lab that has to be set up or any a manufacturing unit that manufactures these kinds of kits has to have not only industrial approval, but also they should state what kind of work they're doing, what kind of product they're making, and these authorities should definitely go look at that.
so don't make those tools accessible and don't set up labs without oversight
Yes.
and without proper regulatory
And
approval.
the most important thing is, as I mean, even the teaching community should probably make their students aware of what is okay and not, which kind of curiosity is not okay.
Right. There's a question here on whether GMOs can trigger extinction of some species. Do you see that possibility?
Yes, yes. So some of these modifications, if it is going to give it a benefit as a population, for example. So, there was some thing where they had gene edited a Anopheles mosquito, which would not uh, allow malarial parasite to grow in it. So suppose this would be selected by nature, this would dominate, and that whole species can be replaced.
So one other question: in nature, can you ever see BT gene jumping into a plant by itself?
I have my own fears about that. I had been actually trying to see if COVID-19 was one of these extra chromosomal DNA proteins, pieces of genetic material that became a virus. This is one theory that some actually I also have, and some of the virologists also have this in their mind, because nobody knows how viruses came into existence. So some of them actually believe that viruses were these extra chromosomal pieces of genetic material which became infective. So we don't know. Anything is possible.
Are you aware of any labeling regimes that are required for gene edited products?
No. There are no such regulations, is what I understand, because many of them haven't even gone for approvals and they've directly let out their products. From some of these sites that whose links I have shared, if you go and see, there they have a list of things that weren't even approved and then they were in the market.
Right. One last question. I think we will close with this. Are viruses mutated or are they just malformed bacteria?
No, no, they are not bacteria. They just have genetic material, DNA or RNA, and a protein coat. That's it. So these viruses can get into any organism, insert their gene into that organism, make that organism produce copies of their own thing. They are not living, actually. Viruses are not living things, but they can use any living thing to make their copies of their own, stuff, genetic material as well as protein.
Right. Um, do you want to end this by anything that you want to say for the next minute or so? And then we'll just see everyone, say bye to each other, and then plan whether we need one more session as a follow-up session or maybe for a completely new batch of people. Um, one
Yeah,
second, Kritika, there's
yeah.
what are the
Some
views of there is a question. Sorry, I have to
Yeah,
ask.
no, somebody had asked about antibiotic resistance also. So I just thought I'll mention about that.
Yeah.
I was working in a lab in Chandigarh, which was working about, deciphering how antibiotic resistance is, acquired in a bacterium. And they found that it has a pump on the bacterial cell surface. As soon as the antibiotic comes in, it will detect and pump it out. So this was what that lab discovered. But after that whole procedure, whatever organism we used for these kinds of experiments were let out, were autoclaved and let out. While autoclaving, only the bacteria will die, the gene will remain. So this is how, so this is the thing. Suppose I had 100 bacterial cells in that particular test tube and I autoclaved it. Even if one bacterium was alive, it was sufficient for that resistance gene to get away. So this is why I have been fighting against waste management, because as scientists, it's more important that we have this concern about not harming nature. By just trying to understand something, we shouldn't be meddling with it, is what my concern is. So this thing I keep repeating everywhere, but then I love it. So as teachers, or as please take this message from me as a closing message. As long as science was asking what, why and how, it was a very beautiful place, or um, place to be. But when the questions became what if and why not, we are getting into dangerous things. So we should never be looking at what if and why not. And in fact, last time, I think Sridhar sir was saying, people now started asking, so what? So let us not get into those questions, is my only concern as a scientist, as a science lover.
Right. There is a question that you may want to end with. What are the views, as you can assess it, about the larger scientific community about gene editing? Are they mostly gung-ho about it, enthusiastic about it? Are they aware of caution that needs to be exercised?
So this actually, when we began, before everybody was had been allowed in the meeting, I had actually addressed this. Some of these people who actually developed this CRISPR-Cas technology, these three people, if you just Google up their name and look into the YouTube video that they present their work, they are very, very proud of their work. So that is why I say, when I was in the field also, I used to become this, horse with just that view. My experiment is working. I have got this result. Oh wow, this is going to make big change in the world. Would only be the viewpoint of a scientist, I think. The moment I stepped out and looked at the whole thing as a third person, I was concerned. This I'm able to say with a little more confidence, because when I took this concern about waste management in an international conference in Calcutta, there was this Asian Crystallographers Association conference where somebody was presenting how they developed a vaccine against SARS. So that time he was saying what all mutations he tried to get the exact fit for an antibiotic. So, antibody, which could be developed as a vaccine, which means he himself has tried all those mutations and left it out in the open. So they didn't even wait for the virus to evolve. All the tools we have supplied on our own. So when this concern came in, I went and spoke to that particular scientist and head of Australian Crystallography Association. There was one lady who was also the vice president of Crystallographers Association. When I went and spoke to her about this particular concern, then she was like, oh my God, we never thought of this. So there are these things that scientists, when they're looking at one particular work that they do, they are probably not looking a little with a broader aspect.
Right. As you can see in the chat box, there are raving reviews of your
Okay.
presentation, your whole session. People are extremely thankful and happy. And we've all benefited tremendously because one, with your entire selection of graphics and videos, you made it visually representative for us. And you brought in so many commonplace analogies for lay people like me to understand. It was very useful. I thank you on behalf of all the participants.